As you know, all cells are enclosed within what we call a membrane, a cellular membrane, that separates the interior of the cell from outside. But at the same time, you need to exchange the materials between the two sides of the membrane. You need to bring in nutrient molecules and to expel waste molecules. And in order to do that, specialized proteins have been involved, which we call membrane transporters. There are kinds of membrane transport proteins that we call channels. And when you open a channel, you just have an open pathway for substrate, ions, sugar molecules, etc., to go through. Whereas in the case of membrane transporters, which we refer to as active pumps, they cannot be open to both sides of the membrane at the same time. They have to work against this and pump in the opposite direction. And because of this fundamental difference between active transporters and membrane channels, they have to undergo very large conformational changes. You recruit the substrate, you have to close on one side before you open the other side. And that's the only way that you can have directional transport. Experimentally, we cannot describe the entire transition process. So the structure of a molecule, you can still get that from certain experimental approaches, but how it moves is extremely challenging to experimental approaches. We might be able to get the initial and the final states, for example, from X-ray crystallography, but how it goes from the initial state to the final state is completely unknown because you can't reach spatial and temporal resolutions that are needed to describe these motions. And the computational methods are the only way to describe this. The holy grail of structural biology of membrane transporters is to describe this whole transition. Why? Because right now, we are working with only initial and final structures as targets, for example, for drug design. We can only design drugs for this form and this form of the protein, whereas if you know all the intermediates and forms adopted by the protein during the function, then you have many more targets to attack. And the chance of finding a selective drug that doesn't work with the other proteins and doesn't cause side effects is much higher if we know all the intermediates involved in the function of this transporter. What we did in this work was for the first time to describe the motion, the kind of structural change and transition that connects these two dots through our computational simulation. What we do is we start with the initial structure of the protein, and then we push the protein along different pathways to go from the initial state to the final state. And then along each pathway, we can measure how hard it is for the protein and compare this pathway to see what pathway might be the most relevant pathway. So in order to act as a pump, you need to, of course, have a source of energy. You need to have some sort of fuel. The cell doesn't have access to very high energies. All cellular processes just use a few kilocalories per mole of energy. And in order to find how it happens really in a cell, in nature, we have to identify pathways that require really low amounts of energy. The way these calculations function is that we calculate interaction between individual atoms in a molecule, and we have to do this for all pairs of atoms. And if you can imagine, if your molecular system includes several 100,000 atoms, you have a lot of calculations to do. One of the main outcomes of our work was that it's actually very difficult for the protein to directly, in a linear fashion, go to its final state. It has to take many turns during its function, and it turns out by allowing the protein to take a more complex transition, we can find much better pathways that are most likely relevant to what happens in nature. It's almost like a dance rather than a simple move within the protein structure. So you have a set of coordinated motions. The order of these motions were not known before our simulations, but we are showing here that the protein is most compliant if you do these stages and moves in certain order. And that fits and explains the overall mechanism of the protein. So in this paper, we looked at an ABC transporter. A prominent member of this family is p-glycoprotein, which is a major protein involved in multidrug resistance in cancer cells. They essentially pump drugs outside the cell, and that's a mechanism of developing resistance against anti-cancer drugs. If we can find out how to block these proteins, we might be able to have a much better penetration of drugs into cancer cells, for example.